HIV pathogenesis. These two questions were posed in the second half of the last century when this epidemic or pandemic started to be recognized. Maybe the world started to ask the second question before the first, as this syndrome was noticed in 1980 and the HIV was isolated in 1983. So in these 3 years, all the world was asking, from where did this virus come? Where did this syndrome come? And in these 3 years, many other viruses were implicated as they are the cause of the syndrome, like human herpes virus 6, cytomegalovirus, all this until the HIV was isolated in 1983. In this sentence, I'd like to say that nature has a big global reservoir of viruses. We have a very big global reservoir of viruses in nature, and it was supposed that this reservoir is highly species specific. We all know that viruses are species specific, and also they have special tissue tropism. So we have animal viruses, plant viruses, insect viruses. All the species has their special global reservoir of the viruses. So HIV was there in nature somewhere. It was new to humans, but not new to nature. The origin of the HIV was the simian viruses, which were known as the simian immunodeficiency viruses. These are the natural hosts of the simian viruses. An important thing here is that infection with HIV in these simian viruses do not cause immune deficiency. Yeah, the monkey is infected, he seroconverts, or it seroconverts, but it does not acquire the disease. This is a big problem in developing vaccines, one of the big problems in developing vaccines, because we don't have an animal model for vaccines. We don't have an animal model for HIV. When we want to try vaccines, we have to try them on the infected people and not on animals. Viruses, in the last few decades, we knew that viruses can what we call hop species, or jump through species, from one species to the other, what we call viral traffic. The jumping of this from one species to the other is called viral traffic. What makes these viruses jump is first to change the established patterns between the human and animal contact. We change these established patterns. يعني زي ما احنا كنا بنشوف اول ما الايفيان فلو طلع، هو في العاده الناس لما كانت بتجيب الفراخ ولا غير الفراخ بتتربى كده بره، حتى الحتت اللي بيعملوا فيها بتبقى بسلك ومتهويه، لما ابتدوا يعملوا بقى الصور بتاعت الكتاكيت الصغيره، تلاقي الصين مثلا صوبه كده فيها مليون كتكوت وجواهم 10 عيال جوه الصوب دي مع الكتاكيت بيوكلوهم ويشربوهم. This is a change in the pattern of the human-animal host. This helps the virus to try to hop species or jump from one species to the other. Viruses don't have brains, but they have very much genetic intelligence. They are very intelligent genetically. By mutations, by recombinations, reassortments, many things, they can start to adapt to the new host and then cause infection, then cause disease, and then be transmitted to other humans. So these evolutionary pathways that the virus takes when it hops the species from one species to the other is the main cause of the new viruses appearing in humans. And that's how the human immunodeficiency viruses had moved from simians towards humans. So the evolution analysis from the simian viruses started since the 1930s. So it takes a long time for the virus to adapt to the new host and start to cause infection and then starts to cause disease. These maybe are the two main variants of HIV we have in the world, the HIV-1 and HIV-2. The HIV-1 and HIV-2. HIV-1 is the most prevalent virus around the world. It's highly virulent and it is present globally. And the natural host is the chimpanzee. The natural host is the chimpanzee. HIV-2 has a lower virulence. It is still now confined to West Africa, and the national host is the Sooty Mengabili. Whether the chimpanzee or the other, both don't get AIDS when they are infected with the simian immunodeficiency virus. So this is where the HIV as a virus came to humans and hopped from species towards the humans. What about the syndrome itself? Did it jump suddenly from the jungle? No, it didn't jump suddenly from the jungle. It was there in Central Africa, moving in closed societies with a stable level, sporadically, unnoticed, maybe since the 1940s or early 50s. So AIDS itself wasn't that new. What was new is the urban pandemic disease. This is the thing which was new in the 1980s. What made this urban pandemic disease develop are driving forces, the urbanization and all these driving forces that led this virus to expand, become well established in humans, and reach epidemic proportions. So that's when we recognized the AIDS epidemic. The problem in HIV, as we all know, is that the system that we need to combat the infection is the system affected by the virus. This is the problem in HIV. We need the immune system to combat infections, but the HIV tackles this immune system and affects the immune system, so you lose the system you need to help in eradication of the infection. So when we speak on the pathogenesis, and the immune defenses, they are closely linked together. Once the patient is infected, he remains infected for his life. This is facts we have to know about HIV. Once the patient is infected, he remains infected for life. And it's not the importance of being infected, but he is also infectious. He remains infected and he is infectious for life. So once we have an HIV antibody positive patient, so we have to know that this patient is infected, he is infectious, and he will remain infected and infectious for his life. Okay? What are the main cells that are affected with the HIV? T4 lymphocytes is the main target of HIV. Then the monocyte macrophage series. Maybe these are the most important two cells affected by the HIV. Dendritic cells are very important also because dendritic cells and the submucosa of the genital tract mainly, they carry the virus and runs, taking the virus into the lymph nodes, what we call, they act like the shuttle bus that takes the virus from the site of infection and distributes it into the different lymph nodes in the body. HIV is also enteropathic. It affects the intestinal villi and destroys the intestinal villi. As we know, the syndrome, the pathogenesis of the syndrome is divided into two main big arms. The first arm is the deterioration of the immune system, the dysfunction of the immune system. And this dysfunction of the immune system manifests itself in the form of tumors, in the form of infections, whether common infections or opportunistic infections. The second arm is the HIV pathogenesis itself. So the syndrome, or the syndrome, we put it under one of these two arms, either the deficiency in the immune system or the HIV pathogenesis. The HIV isolates in the infected patient, we have entropic and T-tropic isolates. Entropic that attacks mainly the macrophages with the co-receptor which is the CCR5, and the T-tropic lymphocytes, it has its co-receptor which is CXCR4, and it is more pathogenic than the other one. Here are the receptors for the HIV, the CD4 with the co-receptors, and we have to say here that receptors for viruses are not there just to wait for a virus to come and adhere to it. Receptors of viruses on the surface of cells have many metabolic functions in this cell. That if we come and block these receptors, the function of the cell will be altered. The function of the cell will be altered. أنا بقول الكلام ده عشان حاولوا كتير إنهم يسدوا السي دي 4 ده، يقولوا حاجات يقفلوها، يداون ريجوليت السي دي 4، يعملوا حاجات من دي. Now that the functions of these cells are highly altered. The main feature in HIV infection or in the pathogenesis of HIV infection, as we said, is the progressive damage to the immune system, which is due mainly to the depletion of the T helper lymphocytes. T helper cells are affected by HIV qualitatively and quantitatively. Qualitatively, that is to say, they are functionally altered. They are functionally altered. They don't work properly. وفي حاجات كتير بيعملها ال HIV, so it doesn't make the cell work properly. Quantitatively means that the number of cells become reduced. The normal average number of T4 lymphocytes are around 1,000 cells per microliter. Up to 500, this is above the immune deficiency. We have a range to move in from 1,000 to 500. Below 500, we start the immune deficiency. First level up to 300 or 200, and the second level is below that. What are the mechanisms that HIV perform to destroy the T4 cells? This is the main pathogenesis of HIV. These are some of the methods by which HIV destroy the T4 cells. Cytopathic effect, as we all know, the harsh viruses are lethal to cells. They cause lysis of the cell. This is because when the virus enters this cell, through its regulatory proteins, it tells the cell, stop working for yourself and start to work for me. So all the macromolecular properties of the virus, all the macromolecular synthesis of the virus stops. And so when the virus ends its replication, the cell dies. And when the virus starts to bud from the cell, this also destroys the virus, the cell, sorry. Syncytia formation is another problem where the infected cell, we know that the infected cell produces the viral glycoproteins early enough to be situated on the surface of this infected cell. So when we have an infected cell, on its surface, the GP120, this G120 attaches to CD4 receptors of uninfected cells. The T4 lymphocytes which is uninfected, has CD4 receptors on its surface. The infected cell, early enough when the virus enters into this cell, it produces the viral glycoproteins and expresses it on the surface. So these viral glycoproteins attach to CD4 receptors of uninfected cells, producing syncytia, and by this way, one infected cell can destroy 10 or 15 cells uninfected around it. So one infected cell can destroy 10 or 15 cells of uninfected cells around it, and these syncytia are short-lived. Cytotoxic cytotoxic T cells. This is the earlier adaptive immune response that takes place, and it is the cause of the drop in the level of viremia once the immune response takes place. And we see here that the infected cell produces its viral peptides on the surface with the class 1 histocompatibility molecule, and then the cytotoxic T cell with the T receptor together with the CD8 antigen attacks the infected cell with the perforin and granzymes and destroy this infected cell. Of course, this is a good immunological response, but it is not enough to clear the infection. Here we can see how the memory clone is not produced in infected cells. This is a resting T cell. The resting T cell, normally, it is resting once the antigen-presenting cell comes with the interleukin-1 to stimulate this resting T cell. It becomes immunologically activated, producing interleukin-2 receptors, and the interleukin-2, they attach to each other, and the T cell replicates or multiplies to produce a clone of cells, maybe 1,000 cells from one activation, and these clones are ready to attack the antigen when it comes again. So we have a store of ready memory clone cells to attack any infections coming again. The HIV infected cell, we get the provirus in the infected T cell, and the T cell, when it is resting, not activated, mostly the provirus is latent. Mostly the provirus is latent if the T cell is not activated. That's why when we get a patient who is HIV positive in the clinical latency stage, we usually tell him as much as he can not to stimulate T cells. No drinking, no smoking, don't get ill. If she's a lady, no pregnancy, no. Many no's, so that as much as we can, we keep the T cell away from any antigenic stimulation. Because when this T cell becomes stimulated, all the genes are turned on with the provirus inside the cell. It replicates, the cell dies, and the memory clone is a stunted memory clone of 2 or 3 cells only instead of the normal healthy 1,000 memory clones. So even when we need to take T4 cells from the store, we don't find. مخزن فاضي, مفيش. Custard, and the store is empty. No memory clones due to infection with the. As we said, the replication occurs in dividing or stimulated, antigenically stimulated T4 cells. Resting cells carry the virus in more or less latent state. Once you stimulate the T4 cell, it turns to be an AIDS virus factory producing a lot of viruses. The problem is that the T helper lymphocyte, we always call it the maestro of the immune system. It controls with the inductive signals it produces and the lymphocytes all the different immune systems. So when the T helper lymphocyte falls, we get a global fall at the end of the immune system. The T cytotoxic, the memory clone as we said, the suppressor, the B cells, all the immune system falls at the end. It starts by the cellular immunity, but at the end there is a global destruction of all the immune system. I just here want to draw the attention that infection is not only in lymphocytes, but we have a great bulk of mucosal lymphoid tissue in the gastrointestinal tract. And this mucosal lymphoid tissue in the gastrointestinal tract is also affected greatly by means of the HIV. We get a drop of this mucosal lymphoid tissue. It rebounds again, but still remains beyond the normal, and this produces a problem in the mucosal barrier of the intestine. We get microbial translocations, we get antigenic stimulations through the intestinal tract, and this makes continuous immune activation for the system. Continuous immune activation for the system. Just to put in our mind that the lymphocytes is not only present in lymph nodes, but they are also present abundantly in the gastrointestinal lymphoid tissue, and this is affected early and remains affected throughout the course of HIV infection. The second important cell to be affected with HIV is the monocyte macrophage series, which plays a major role in the pathogenesis of HIV. But the monocyte and macrophage is affected only qualitatively. It is functionally altered, but it is not affected quantitatively. That is, the virus is not lytic to the macrophage, and the number of macrophages remain the same. The macrophage is one of the greatest reservoirs of HIV in the body. It is one of the greatest reservoirs of HIV in the body. So the macrophage and the resting T4 cells are reservoirs of HIV in the body. Also, the macrophage is the cell that takes the virus across the blood-brain barrier and make it reach the brains or the nervous system. Also, it is the main cell that makes the HIV reaches the lungs. This is well known for the students, which is this triangle that starts with a broad base after the acute retroviral syndrome, in which the viremia is very high. The viremia is this straight lines. The viremia is very high in the acute retroviral syndrome. The patient doesn't show any symptoms except some vague symptoms like some sweats, some fever, some infectious mononucleosis. It passes unnoticed. Most of the time, this acute retroviral syndrome, the patient is still antibody negative because the seroconversion in these chronic cases takes a long time, maybe 3 weeks, 3 months. It may reach up to 6 months. Once the immune system starts to cause the immune response, we find that the immune response drops the viral load or drops the concentration of the virus in the body to low levels. But unfortunately, it can't clear the virus from the body. بني ادم بيخف ازاي؟ بيخف لما الاميون سيستم يطلع يقضي على الفيروس، ما تلاقي فيرال كليرنس، خلاص يبتدي البني ادم يخف ويخش في الكونفاليسنس. لكن لما الاميون سيستم ما يقدرش يعمل فيرال كليرنس ويسيب شويه فيروسز in the body, then we will move to the chronic stage. After the immune response, وبعد ما يحصل immune response والفيرال تيتر يقل، بخش في stage of clinical latency, or what we call them people living with HIV. These are completely normal people, going and coming completely normal. They don't have anything to, they don't have symptoms, they don't have signs, they are just HIV positive. ولو نلاحظ احنا بنقول هنا clinical latency، ما بنقولش virological latency, because the virus in the body is always replicating. The body as a whole is in a low-level chronicity state. The virus is always replicating. The virus could always be isolated from the body, so that's why we call it clinical latency. No symptoms, no signs, but not virological latency, okay? That's why clinical latency, as the virus replicates, we see that the CD4 slope starts to drop. Drops by years. Maybe 50 to 100 cells per year. This is the CD4 slope. So the CD4, as it drops, the viral load starts to rise, and when they meet, this is the first level of immune deficiency, and we enter into the AIDS-related complex, or the chronic constitutional disease, or what we call the symptomatic HIV infection. The viral load is a predictive marker for long-term progression of the disease in years. لما الاميون سيستم يقلل الفيرال لود قوي يبقى البروجريشن احسن. لو قل الفيرال لود مش قوي يبقى البروجريشن اقل. So the viral load is an important predictive marker for long-term progression of the disease. The CD4 count is a predictive marker for short-term progression of the disease, يعني شهور, mainly to the opportunistic infections. Continuing the slope of the CD4 count, we move from the AIDS-related complex towards the frank AIDS. AIDS-related complex has 5 main signs and symptoms: fever, chronic diarrhea, loss of weight, skin manifestations, and common infections. 5 دول هما اللي بيبقوا موجودين في symptomatic stage: fever, chronic diarrhea, wasting syndrome, skin manifestations, and the common infection. The common infections come severe, extensive, responds poorly to treatment, لأنه طبعا البني آدم immune deficient مش زي العادي. If the CD4 count falls below 200, then we enter into frank AIDS. What is added on the frank AIDS is the tumors, mainly the Kaposi's sarcoma, the nervous system manifestations, opportunistic infections that don't come to normal people, and this is the general manifestations of the AIDS. Persistent generalized lymphadenopathy can be present either with the clinical latency stage or with the first symptomatic stage, and this is due to reactive hyperplasia of the nodes when the virus enters these nodes to replicate. The nodes cause reactive hyperplasia and become enlarged. When the disease progresses, these nodes collapse. The patient comes and he is happy that the lymph nodes are disappearing, but this is a very bad sign in the progression of the disease. This means that the lymph nodes cannot anymore harbor this virus, and the architecture is exposed, and the virus will pour in the circulation, and we will get a very high titer, and the progression of the disease will continue. Okay? At the beginning of this century, scientists found that this dark line is the average of all the HIV infected people, and the course on average without treatment takes around from 10, 12, 14 years maximum from the base to the tip of the triangle. A very small amount, maybe 2 to 3%, they are rapid progressors. All the course ends in 2 to 3 years. We don't care about these, but what the scientists looked for are the 2 or 3% which they call the long-term non-progressors. What do we mean by long-term non-progressors? That is, the patient remained for 20 years in the clinical latency stage and he did not progress towards the symptomatic stage. دول a long-term non-progressor. عدى عليهم 20 سنة هم infected and they didn't progress towards the symptomatic stage. طبعا الساينتست مسكوا كم واحد دول بقى فلوهم، مش عرفوا ايه السبب، اشمعنى دول فيهم ايه علشان يبقوا long-term non-progressive. لقوا ان co-receptors and the HLA polymorphism plays a role in this long-term non-progressive. The genes of the co-receptors, the CCR5 or the CXCR4, when we have mutations in these genes, then the patient may take longer to progress to the symptomatic stage. Also, some of the HLA alleles, like HLA that enter in most of the infections, some of the actually alleles, if they are present in the patient, this makes him a long-term non-progressor. They started working on these co-receptors, but still we don't have any answers for these. These are just a bit of lucky people that they had some mutations in their co-receptors that made them stand infection for more than 20 years. And up till now, in spite of the great scientific progression, the disease still outruns the science. We don't have vaccines. Treatment is just to ameliorate or diminish progression of the disease. We don't have any cure. We hope that by time God gives us less virulent HIV strains, that we can stand its infections. Thank you.